human beings, marvelously complex and wonderful biological machines. Like all machines, they are built from a blueprint. Our blueprint resides in our DNA, a structure that contains all the instructions for creating human life. Human beings are created when the DNA from two parents combine, setting off a chain reaction that instructs cells how to specialize and replicate. Before long, heart cells, lung cells, blood cells, skin cells, and many other specialized cells form, replicating and developing into all the pieces necessary to create a human. While humans are not unique in this type of genetic construction, they are wondrously complex, and unraveling the blueprint of humanity was a tall task. While early biological discoveries, such as RNA by Friedrich Miescher in 1886 and DNA by James Watson and Francis Crick in 1953, paved the way for sequencing the human genome, it wasn't until 2007 that the code was cracked, and the complete human genome sequence was published, all 6 billion letters of it.
Publishing of the human genome was a big deal because it was a monumental step for scientific discovery. It showed the world what could be done through scientific collaboration and advances in technology. What that led to wasn't just the field of synthetic biology, but advanced scientific progress as a whole. You had advances in cancer research, in drug discovery, genomic databases, all of that sprung from the Human Genome Project.
With the publication of the human genome, synthetic biology began in earnest. 3 years later, in 2010, a team led by J. Craig Ventnor became the first to successfully create synthetic life when they introduced the DNA from a bacterium genome into another cell. At the same time, NASA was becoming keenly interested in advances in synthetic biology, and in the fall of 2010, the agency launched the NASA Synthetic Biology Initiative. With synthetic biology holding such great promise for the future of human space exploration, NASA began expanding its capabilities for conducting research. Working in collaboration with the University Affiliated Research Center, or UARC, NASA began assembling what today is known as the Innovation Lab at NASA's Ames Research Center. This state-of-the-art facility provides all the necessary capabilities to conduct research across a wide spectrum of synthetic biology topics. One such area of research is in light-activated proteins.
So light-activated proteins are basically solar-powered little nanomachines, but instead of taking the energy from the sun and converting it into, say, electricity, it takes that energy and it converts it into motion on the nanoscale. Think about bacteria as little factories that you can engineer to do certain things you want. Now imagine you want to control that factory. Well, what we're trying to do is take light and use it kind of as a way to remote control the bacteria so we can direct them and tell them when we want them to do something. One of the benefits it will provide is real-time control of bacteria, so using these bacteria in a way that nobody's ever been able to use them before. What I'm trying to accomplish and achieve right now is help NASA be able to colonize other planets. So when we get to other planets, what we need is resources to be able to build structures. Now, bringing all the materials needed to build structures is very difficult because it weighs a lot and we don't know necessarily how those things will react in the Martian environment. So what we want to do is develop things here on Earth then that we could use over there. So the system that I'm developing will basically allow astronauts or people who travel to other planets to build specific structures by using the bacteria and proteins to 3D print using the soils.
While work in the area of light-activated proteins is laying the foundation for future advancements, other research at the Innovation Lab has more immediate application. In the area of life support systems, work on regenerative filters may help reduce costs associated with water filtration in the International Space Station, as well as NASA spacecraft currently in development.
If you can think about long-duration space flights, we have only so much payload that we bring up with us, and instead of creating linear systems, we have to create cyclical systems where we're able to process the waste and create inputs into other systems. So what this forces us to do is it forces us to think of creative solutions for filtering water, for converting organic matter, for conserving resources. Waste management currently on the space station involves a lot of outgassing. So what is constantly produced is waste carbons in the form of CO2 in the form of organic matter that either has to be compacted and shipped back down to Earth or released out into space. What that actually entails is a lot of wasted carbon that could be used for other purposes, such as reproducing food, producing filtration systems, or purifying water. So the work that we're doing with regenerative filters is that we're creating filters from biological material. So we are using synthetic organisms to generate lipid filters that act as osmotic membranes. So what they do is they passively filter water. And these membranes, they oftentimes degrade, and once they degrade, they're no longer usable. However, with regenerative filters, we're able to constantly produce these osmotic agents that are able to then constantly regenerate that membrane, creating for longer lifespans for that membrane. We're able to increase the lifespan of these filters, and it minimizes payloads, and it actually promotes long-duration space flights as well.
In addition to regenerative filters, NASA is also using synthetic biology to completely reimagine how human waste can be recycled in space, looking at ways to evolve it from a physiochemical process to a biological one, with the main goals of reducing solids, retaining water, and creating energy.
Current systems rely on physicochemical processes, meaning there's a lot of physical filtration and chemical treatment of wastes, whereas our bioelectrochemical system uses microorganisms to naturally process the waste. This is beneficial because microorganisms can reproduce, whereas if you're using a filter or a chemical added to your system, you have to constantly maintain those or add them. All we need to grow microorganisms is food and a hospitable environment. So the hospitable environment is our microbial fuel cell or bioelectrochemical system, and the food is actually the urine.
While synthetic biology has a myriad of uses for treating waste, both in the near and the longer term, it also has the potential to help us answer one of the most thought-provoking questions in the universe: where did humans come from?
All the life on this planet came from a single common ancestor around 3.8 billion years ago. And it's actually amazing to know that already at that time it had a DNA and the protein and all the systems so that they can replicate themselves. What we don't know is beyond that. We don't have any more genetic information beyond 3.8 billion years ago. So there was something happening between 4.1 billion years ago when the ocean cooled down to the 3.8 billion years ago where life as we know emerged. And that's where the astrobiology kind of kicks in. We want to understand how these simple organic molecules, which were brought from the space, formed into a system that can actually do all the metabolism and harvest energy from the environment and do a self-replication. If you look into the composition of meteorites, especially the carbonaceous meteorites, you actually find many amino acids in these meteorites. And our body is made out of protein and water, but a protein is a chain of amino acids. Life uses 20 common set of amino acids. But out of these 20, actually 10 of them are actually found from these carbonaceous meteorites. We know that there are certain concentration amino acids already present in the early Earth environment. So what I'm actually trying to do in the lab is to actually recreate a protein that only uses these limited set of amino acids that are present in the early Earth environment. What I'm trying to do is to actually create a set of DNA sequence that encodes for these peptides. So actually I'm not making a protein straight from amino acids, but rather I'm using a modern life system to make these proteins. So starting off with a DNA sequence. So this technique is very useful because we can incorporate new genetic material into other organisms, or we could actually design our own new DNA sequence or a protein that can have a completely new function.
From gene therapies that keep astronauts safe and healthy to advanced life support systems, and even the origins of life on Earth, researchers at the University-affiliated Research Center and NASA are breaking new ground in advancing bioengineering and synthetic biology, working to create a future where humans are far more adaptable to a wider variety of habitats and harsh environments, both in space and here on Earth.